All right, so today we're going to talk about the urinary system. And like we've done with every other system, we'll start by looking at, what are the functions? What are the things that it's actually doing for you? And it's doing a lot more than you might think at first if you haven't really looked at the urinary system before. So what are functions of the urinary system?
Removes waste product.
Yeah, so that one, most people can think about, eliminates wastes. So you have a variety of wastes that happen as part of byproduct of metabolism, like urea from breaking down proteins, creatinine. You have, just the metabolic byproducts of drugs and this and that. So things that are in your body that you want to get rid of can get contributed to the urine and then leave your body. They call it excretion, excreted, it leaves your body. So what's another function of it besides that?
Regulate blood pressure.
So it actually is going to be, and it's going to help regulate blood pressure in a couple of different ways. So I'm going to start a little more general, and then I'll talk a little more about how blood pressure is connected. But so it is critical for water balance. So the kidneys are the things, their water balance basically means, you think about, this is you, let's say this is making you as a funnel head. This is you. And then you've also got like an outlet that's leaking out. So there's constantly water coming in. There's water leaking out. And there is a kind of ideal amount of water that's part of your body so that everything is the right concentration. It's usually about 2.5 liters coming in, 2.5 liters leaving every day. And the net effect is everything stays balanced. But the kidneys are the ones that are determining how much water do we have to leave or hold on to in order to make sure things stay balanced. If more water is coming in, the kidneys are going to release more water as urine. If less water is coming in, you're getting more dehydrated, the kidneys are going to keep that water in your body. You'll have much less volume of urine coming out. So the kidneys are critical for doing water balance, kind of adjusting your urine output based on the water input. So the water stays, you don't inflate or shrivel up. And this water balance is also a critical part of your blood pressure control. I think I've mentioned this before, but basically if you think about your heart pushing the blood out into your system, I'm doing one circuit, obviously there's 2 circuits, but it's basically this closed system full of blood. And there is an ideal amount of blood where it has just the right amount of pressure inside that amount of volume of blood vessels. You can, in the short term, you can make adjustments to adjust your blood pressure by changing the diameter of vessels or changing the rate of your heartbeat and stuff. But the main set point of your resting blood pressure is actually the volume of your blood, which is, directly related to the water balance. So the kidneys are actually at the core of long-term control of your blood pressure by regulating water, which regulates blood volume. We'll also see the kidneys do other things to do blood pressure regulation as well. There's this little place as part of the kidney called the juxtaglomerular apparatus that is monitoring your blood pressure and will activate if your blood pressure is dropping, to do all sorts of things to bring your blood pressure back up. So the kidneys are, critically involved And that's good. We're going to see why that makes sense. The kidney function relies on adequate blood pressure. If the blood pressure drops too low, the kidneys will shut down. All right. What else do they do?
It balances like electrodes.
Say it one more time.
Would it balance, does the system balance like electrolytes then?
Yeah, exactly. Exactly. Yeah, no, I didn't hear you because there was like a glitch in the sound. So electrolyte balance is also really important. You know, all of those action potentials, those electrical signals that are coordinating your muscle excitability, or the action potentials going down neurons that are all of the kind of information traveling through your nervous system. It all relies on the appropriate concentration of sodium ions and potassium ions and chloride ions in your body. And if those ionic concentrations aren't right, then the voltages get all weird and you're going to die of a heart attack or something. So electrolyte balance, and it's the same thing, kind of what here I was talking about water balance. Like the kidneys are going to have to adjust the amount of volume of urine based on the volume of water coming in. The kidneys are going to also adjust the amount of salt in your urine based on how much salt is coming in. If you have a lot of salt, you're eating a whole bunch of pretzels all the time, you're going to be peeing out a lot more salt. Your concentrated urine with lots of salt dissolved in it. If you are, on the other hand, just eating carrots and there's not much salt and your body's desperately trying to hold on to the salt it has, there's not going to be much salt in the urine. It'll be very watery urine because you're going to have to, the kidneys are going to make their adjustments based on what the body needs to do to maintain the electrolyte balance as well.
And
Another
also
thing,
temperature control.
no temperature that they don't really do in any way that I know of.
Okay.
The cardiovascular system is involved with temperature by sending more or less blood to the surface of your body. But,
Yeah, and maybe I just when I used to work outside all the time, and so it was a lot of talk about like temperature control based on how much you're peeing or not, and so that's where I was going.
So I don't know about that, but it could just be something I'm not aware of. If you find something about that, I'd be interested.
Okay, I'll let you know.
Okay. Let's see. I'm going to put in pH balance here. This is kind of more of a physiology thing, but you know, there is a homeostatic pH in your body of around 7.4. And if the pH in your body gets too high or too low, you're gonna go into acidosis, alkalosis, or if it gets too far, you're gonna die. And the kidneys are the main long-term control of pH balance. If you have too much, if you're getting too acidic, you actually pee out more of the acids, like the hydrogen ions. If you're getting too alkaline, you can pee out like bicarbonate, alkaline molecules. So, the pH of your urine, is urine acidic or alkaline? It depends. It depends on what you've been eating and what the kidneys need to excrete in order to maintain your pH balance. So these are kind of big things that the kidneys are doing. Some of the things more on the side, RBC regulation, production regulation. So your kidneys make this hormone called erythropoietin, which basically tells your bone marrow to up the production of red blood cells. So like if your blood is not having adequate capacity to carry oxygen around, the kidneys actually release this erythropoietin, which increases the number of red blood cells in your body. Right, that's when people do high altitude training for like a race or something. They go up to a high altitude where the air is thinner, where there's less oxygen coming into their system through their lungs, in which case the kidneys are going to release erythropoietin and increase the amount of red blood cells in their body to increase the ability to carry what oxygen there is around. And then when they get back down to sea level, they're supercharged. Sometimes people use this as a performance enhancing drug, like EPO is erythropoietin. And, to the same effect, you get an increased oxygen carrying capacity of your blood. If you overdo it, you can get into a lot of trouble because, too many red blood cells, your blood's gonna become very viscous. It'll be like kind of having sludge in your vessels and it won't flow well. So kidneys are also actually necessary to activate vitamin D. Vitamin D is made, the precursors are made in your dermis. Vitamin D, I'm going to say vitamin D activation. Vitamin D comes from dermis. It's it's made in the dermis of your skin, and it needs the presence of ultraviolet light. But vitamin D is activated actually by the kidneys. And then this is going to be necessary to absorb calcium from your diet. So kidneys are actually, they're involved with lots of different things. This is a weird, I'm gonna have to figure out a better way to draw this. This is a weird looking picture.
Would it help regulate phosphorus too?
Say it again, Nikki.
Would it help regulate phosphorus too, or just the calcium?
No, it regulates all sorts of minerals. When I say electrolyte, it's kind of all sorts of ions. There are other hormones and things like that are going to work. But and we're going to talk about what's happening in here in a few moments. All right, so what we're going to do now is we're going to start with kind of the big picture and look at, I'm going to draw the kidneys and we'll assume they're doing all this stuff. And then we'll kind of go into a more microscopic view and look what's going on in the kidneys that's actually doing all of this. So we'll start with the big picture. So here's your abdominopelvic cavity. Kind of in your lumbar region. And they're retroperitoneal. So again, if you were doing your rats, you would have to peel back the parietal peritoneum before you actually see these underneath there against the back body wall. They are enveloped by a fibrous capsule. So there's a renal capsule. Whoops. Which is kind of your classic dense CT. And then they actually are in this adipose capsule. It's a major, again, if we had in the cadaver, when you even once you peel back the peritoneum and you look for the kidneys, all you see is this kind of fatty pillow, this pillow of fat. And then you slit open this pillow of fat and you'll see, oh, there's a kidney sitting inside the pillow of fat. And then there's even more, there's more dense CT, they call the renal fascia, which kind of anchor that to the body wall. You know, and then as these kidneys are doing all this stuff, obviously they have a blood supply. What do we call the arteries coming in?
Renal artery.
Exactly, the renal arteries. And a huge amount of your blood flow is routed through the kidneys, way more than the kidneys need just for their metabolic needs. A quarter to 20 to 25% of your entire blood flow ends up going through the kidneys. So like 20 to 25% of your cardiac output, they call it. So that's a lot of your blood. So it's going to be necessary to process and do all that stuff we've been talking about. So a huge amount of your blood is going into the kidneys and obviously getting drained out the renal veins. So I won't put those there in order to make my life easier. The kidneys do all their processing. The stuff that's going to leave is called the urine, and it's going to come out the ureters. And we have a nice little option to not just pee if the urine's getting formed. We can actually store it till we're ready to pee. Duh. And that's because we got a bladder, urinary bladder. And then there's the tube that takes you out of the bladder just to outside, and that's the urethra. Yeah, I'm drawing like a female here. And the female, the urethra is only like 1.5 inches long. If this was a guy, the urethra is going to be a lot longer because it's going to go all along the length of the penis. So we'll be looking at the urethra in more detail when we do the reproductive system as well, particularly in the guy, because the urethra is not just for peeing. In the guy, it's going to be where you mix up all the components to make the semen and send them up into the female reproductive tract. So urinary bladder and the ureters, what kind of tissue lines them?
Ciliated urostratified clothing.
No, that's when you're trying to deal with dust, like in the respiratory system.
Is it transitional?
It's transitional. The stuff that's good at stretching. So the bladder can stretch. It can fill up to like a couple of 100 mils without it feeling anything, but it can stretch and hold up to 1 liter or so. So it's kind of designed so it can stretch. And the epithelium is that transitional epithelium, which can stretch out. Both the ureters and the urinary bladder have smooth muscle in the wall as well. For the ureters, it's kind of, there's some peristalsis to kind of keep the urine moving down towards the bladder. The urinary bladder has smooth muscle. It has a name. I'll give it to you. It won't be on your test, but I'll give it to you. The detrusor muscle. This is just the smooth muscle in the wall of the bladder. And then the outside of the bladder is just adventitia. It's just dense connective tissue that just anchors it on the front of your pelvic cavity there. Other things we should put in here. So the sphincters are just like the sphincters we saw in the anal sphincters, except now they're urinary sphincters. So we're going to have an internal sphincter. I'll type it to make my life easier. Oh, I got to turn on my internal urethral sphincter. This is going to be smooth muscle. So this is going to be the one that's involuntary. And there's going to be another one, and that's going to be my external urethral sphincter. That's going to be skeletal muscle. So that's going to be voluntary. And the basic idea here is just like it was in the digestive system. As the urinary bladder fills with more and more urine, it starts stretching out. That stretching out triggers a reflex that relaxes the internal sphincter, which pushes pressure down a little lower, and that's where you have the urge to pee. And then, fortunately, you've got the external urethral sphincter, which is voluntary skeletal muscle. So you get to choose whether or not this is the appropriate time or place. So that is that. What else? We can talk about the trigone. It's probably good to do that. There's like a triangular region that's defined by where the ureters enter and where the urethra exits. It's called the trigone. And it's kind of it's a bit of a, like a fossa, depressed. It's where you often get urinary tract infections kind of setting up camp. Women in general are way more vulnerable to urinary tract infections than guys because the urethra is so short and it doesn't take that far to get up into the bladder here. Again, for a woman, it's only like about 1.5 inches. And for a guy, again, this is going to be like more like 8 inches because it's got to go through the pelvic floor, all along the shaft of the penis and all that. And the other thing, the urethral opening for a woman is, right near where the anus is as well. So there's more likely to get cross-contamination with like fecal bacteria. So just in general, urinary tract infections are way more common for women. What else do we want to say here? This adipose kind of cushions and supports the kidneys here. One of the things that can happen when somebody loses too much weight, somebody's like really kind of getting more anorexic, you can actually run into trouble because this adipose, usually even in skinny people, there's quite a bit of adipose there to kind of cushion and support the kidney. But if it does start getting reduced, you can actually have the whole kidney droop. The word for drooping is ptosis. And you can, if your eyelid is, the muscle to your eye is, or the nerve to your eye is messed up and your eyelid droops, they call that ptosis as well. Ptosis just means drooping. But if the kidney kind of droops, it can kink the ureter and make it hard for the flow. So it's, you don't want to overdo like, having no fat. So that is this big picture here. Trying to think of other things to talk about for the big picture. No, I think what we're going to do now is go into the kind of kind of focus on what is actually going on in a kidney. What does a kidney look like? What are the processes happening? What are the little microscopic structures that are doing it? So, kidney. Okay. So it's shaped it's kidney shaped, like kind of like a kidney shaped pool or something. It's got an outer part. What do we call the outer part of anything?
Cortex.
Exactly. This is going to be the renal cortex. And then this inner part is going to be the renal medulla. And when you look at the renal medulla, you have these regions. They're kind of triangular. We actually do have on one of the videos I have, you can actually see they slice open one of the kidneys. These are called pyramids, renal pyramids. We'll talk about exactly what they are. It's basically where all of these ducts are going through that with, helping process the urine. The space in between the pyramids, they call the columns. These are all ultimately going to pour out into this system of little ducts and ultimately become the ureter. So if this is my ureter, the ureter, which is going to leave the body, and that should make sure you remember, be sure you're careful about distinguishing urethra. Urethra goes from the bladder to outside. Urethra is the thing that leaves the body. Ureters, on the other hand, are going from the kidneys to the bladder. So they sound kind of similar, but they're different parts of the journey for the pee. So we're looking at the ureters now. We're looking at this point here where the urine is formed and being sent on its way to the bladder. This area where all these little pyramids are dumping into, they call this the renal pelvis. These little doohickeys leading into the pelvis, they call those the calyces. And there's major calyxes and minor calyx. Major calyx versus minor calyx. It's probably worth, here, I can share. Let me go. Hold on, I gotta share screen. Yes. Okay, we've got Mr. Complete Anatomy here, and we're gonna turn on his urinary system. Yeah, it's definitely his, isn't it? And here we have the kidneys kind of sitting here in the kind of lumbar. Whoa, I did that again. This lumbar region here. And what I'm going to do is I'm going to cut away half of the kidney, open it up. And here what you're seeing, these are the pyramids. This is, in the cortex and in these pyramids are where you have all these tubules that are doing the processing. We're going to talk about that in more detail later, the nephrons and the collecting ducts. But what I just want you to see right now is this structure here. This is the renal pelvis. It's kind of where all of this finally funnels into before it leaves out the ureter. And then these are the calyces. So minor calyces would be kind of the smaller ones that funnel in, and then the bigger ones that are the convergence of many minor calyces is a major calyx. So that's all a calyx is. A calyx is these branches, these tributaries. The small ones are minor calyces. The big ones are major calyces. Whoops. And ultimately, they are coming into the renal pelvis. Renal pelvis ultimately is heading into the ureter. And let us get rid of this, guys. Okay, I want to get rid of all this right now. I just want the bladder. Okay, We'll see all this again tomorrow. All right. All right, so here is the bladder. Again, I want to just get rid of more of this even. There we go. Okay, so the bladder is here. And here we have the ureter coming down. It starts there, comes in. If we open this puppy up, here you can actually see the trigone. I took too much out here. The trigone is this triangle here. You can see here's where the ureters are entering. Here's where the urethra was exiting. So that's this kind of depressed area in here is the trigone. That is the that little triangle area. And again, we'll come back to the bladder and the urethra when we do the reproductive system. But I think this is kind of nice for showing particularly just this idea of the pyramids and the columns and the calyces and all that. So let's go back to the one other thing about the big picture is you do need to know the names of the arteries that go into supply everything. So let's talk about that. So here's the kidney, here's the cortex, here's the medulla, here's my renal artery coming in. And the little nephrons, the little things that are processing your blood and ultimately creating the urine, all these little nephrons all start here in the cortex. We're going to talk about that in much more detail, but for right now, we're going to talk about how all these 1 million little nephrons, little tubule processing units, are going to get their supply of blood coming off this main hose coming in. So as you kind of enter into the beginning of the kidney, they start branching into somewhere around 5. They call them segmental arteries. And then you get arteries that start going up through the medulla. And these are going to be called interlobar arteries. And then when they get to the boundary between the cortex and the medulla, again, cortex, medulla, they start arcing. And that word arc is going to be helpful to remember their name. They're called arcuate arteries. And then radiating off of the arcuate arteries up into the cortex, again, the name, they're called the cortical radiate arteries. There is another name for them. Sometimes they're called interlobular. But cortical radiate just is so much more descriptive because they're radiating out into the cortex. That's the one I like to use. Right, so, and then coming off of these cortical radiate arteries are going to be the little arteries that feed each of the nephrons, which we're going to look at in more detail in a few moments. So you should, on a test, I could easily like ask you about this though. You should know these. The other thing you should know about the big picture of the kidney before we start getting small is the saltiness of the kidney varies dramatically from the cortex down to the medulla. So when you are in the cortex, the osmolarity, the kind of saltiness is normal. It's the normal saltiness of your body fluids. As you get down into the deep medulla, it's 4 times as salty as it is in normal body fluids. So as we get down into the medulla, it's 4 times saltier. It's not all salt, it's different solutes. The more accurate term would be it's 4 times higher osmolarity. But it means that this is part of the way you're going to be able to reabsorb water because it's really really high osmolarity, high solute concentration, which by osmosis is going to basically pull water back into the body if you allow it. So this high osmolarity, high saltiness of the medulla is going to be critical into the functioning of the kidney as we get deeper into this all. Let's see. It's what I think I'm going to do. I'm going to lay out the basic processes that happen in each of these little tubule structures all along the outside here. And then we'll take a break. We'll finish up with the kind of details of the nephron, and then we'll start lab. So let's let me just go into the basic processes that allow the kidney to do all those things that we were talking about in the very beginning, the water balance, the salt balance, that kind of stuff. So the structural and functional unit of the kidney is the nephron. So there's going to be like a million of these nephrons all working in parallel. So I'm going to draw one nephron, but again, this is happening again and again. Right, so there's going to be this cortical radiate artery coming up. So we're in the cortex. Then coming off of that cortical radiate artery is going to be the little artery that feeds our nephron. This is going to be the afferent arteriole. This is going to be the third example of a portal system that we're going to see. Remember, we had the hepatic portal system where you went from the capillaries of the digestive organs to the capillaries of the liver. We had the hypophyseal portal system where you went from the capillaries of the hypothalamus to the capillaries of the pituitary gland. In this case, we're going to go also from one capillary bed into another. So the first capillary bed we go into is this little twisted ball of capillaries called the glomerulus. And the glomerulus is made out of these really leaky capillaries. Do y'all remember the capillaries that have like the little windows in them?
Sinusoidal capillary.
These are the fenestrated capillaries. So fenestrated capillaries. And fenestrated capillaries are really leaky. So you've got all this kind of stuff leaking out. This is what we're going to call, this is the first step of our whole process. We're going to call filtration. So at this point, filtration, we're going to have water. Let me do it. Water and small solutes, leave the blood. Both good and, both nutrients and wastes. So this is just based on size. So small molecules like urea, which is a waste, those leave, but also small molecules like glucose and amino acids, which you actually want to keep in your body. So it's very indiscriminate in terms of whether it's good or bad. It's just basically about size. Larger things like your blood cells and large plasma proteins, they stay behind. But this filtrate is going to be all sorts of stuff. And not only is it got good stuff, but it's huge amount. I talked about like, a fourth of your blood flow going into your kidneys. Like a tenth of that is becoming filtrate. If you just look at the filtrate, you're making about 180 liters per day. 180 liters, that's a huge amount. That's something like 75 gallons or more of filtrate. You obviously, this is not pee. You'd be dead pretty instantly if this was all leaving your body. So what we need to do is we need to catch it, which is going to be the job of the renal tubule. This first part here we call the Bowman's capsule. So we capture this filtrate, and now what we need to do is we need to recover most of that water and all of the nutrients, the stuff that we want to keep in our body. So that's going to be the job of the second capillary bed. So this Afferent arteriole enters into the glomerulus. The glomerulus then has another arteriole leaving called the efferent arteriole. Right, that's not normal. Normally you have a venule leaving a capillary bed. Here we have an afferent arteriole into the capillary and an efferent arteriole leaving the capillary, which then feeds into this next thing, which is my peritubular capillaries. Let me write that. This is messy. And so we're going to have two major things happening. First thing is the stuff we want to keep in our body, like all of that water, all the nutrients. They're in the tubule, right? They're all just going down. So we have to take them and actively move them back into the bloodstream, back into the body. So that part 2 is going to be called tubular reabsorption. So you recover water and nutrients. When we actually look at the histology of the kidney, we'll see the tubules that are doing this they have a lot of microvilli to increase surface area for reabsorption. It's going to be easy to recognize the tubules that are involved with this heavy duty reabsorption because they have modifications to increase surface area. They'll have really fuzzy lumens when we look at them. And then part 3, if there's stuff that's still in the blood that we'd like to get rid of, maybe there's some metabolites of drugs, or maybe there's still some salts that we wish we were getting rid of. We want to pump out some more sodium to ultimately pee it out, or we need to pump out some acids to adjust our pH. So you have an active way to actually what we call tubular secretion. So this is anything that's in the body that you want to get rid of, you can put in the tube. So by the time it gets to the end, that's going to be like your urine there that's actually just leaving the body. So if there's things you want to get rid of, you put them into this renal tubule before it's all over, and that will ultimately leave your body when you go pee. So any questions about the big picture of this nephron? I had the kidneys. When we talked about renal artery and the segmental artery and the interlobar arteries and the arcuate arteries and the cortical radiate arteries. And coming off of each of those little cortical radiate arteries is going to be that little afferent arteriole feeding into a glomerulus. And then rather than doing a straight little tube like I just showed you right before we took our break, it's actually going to take a very convoluted pathway. There's going to be a proximal convoluted tubule and the descending and ascending limb of the loop of Henle and a distal convoluted tubule, and finally going into a collecting duct, which eventually goes down into the calyces and into the renal pelvis. So that's what we're going to look at now, is this big mess that's the renal tubule. We're just going to focus on the very basics of the functionality and the anatomy of it. In physiology, we go way deeper into the detailed functional properties of each of the parts of the tube. For our purposes, we are going to simplify a little. So the nephron, chapter 2. So a more detailed look. Again, it's still going to be doing filtration, reabsorption, secretion, but there'll be more to see. It'll still start with the afferent arteriole coming in. Into the glomerulus. Now we're going to look a little more detailed at the actual Bowman's capsule, the beginning of the renal tubule. So the renal tubule, basically it has these cells that are more squamous that kind of make kind of the funnel part. And the tubule itself starts out actually cuboidal as we start into the tubule proper. And then there are also cells that glom around the actual capillary cells, the capillaries as well. So the glomerulus, this whole thing is called the Bowman's capsule. And actually, including the glomerulus, altogether, this is called the renal corpuscle. Let's look a little more detail at where this tubule and the capillaries come together. If this is my capillary with the little fenestrations, the cells of the tubule actually have these little fingers that interlock. So they're called podocytes with their little foot processes. They create these little slits. They're called filtration slits. Also called slit pores. So that's what's actually making that filtration membrane. That's what's keeping the blood cells and the large plasma proteins back and keeping them in the bloodstream. And the stuff that can make it through the little fenestrations and slit pores to become the filtrate is the water and very small solutes. Basically anything smaller than like 3 nanometers gets out, anything larger than 7 or 9 nanometers gets held back into the bloodstream. After I'm done with my kind of whiteboard stuff, the book actually has some really nice photo, kind of electron micrographs, kind of showing what these podocytes are like. But basically, it's these cells that have these interlocking finger-like processes that make these little slits that form part of this filtration membrane to help create the filtrate. So that's all happening here. So now we have the filtrate forming. And like I said, 180 liters per day, containing good stuff and bad stuff. So the first major part of the tubule is going to be where we're going to reabsorb a lot of the volume and a lot of the good stuff. And it's going to be a big convoluted tubule called the proximal convoluted tubule. I don't know, I'll just draw it like a big convoluted tubule. So the PCT, which means proximal convoluted tubule. This is a major site of reabsorption. Obviously, the peritubular capillaries have to come back and are going all around here as well, right? So this is where we have stuff that's in the tubule that we want to keep, like maybe water or nutrients, and we pull it back into the bloodstream. That's what that reabsorption is. Something that is in the tubule that was part of the filtrate, but you actually want to keep, you reabsorb it back into the bloodstream. Under the microscope, you are seeing parts of the PCT because it's optimized for reabsorption. When you look at a cross section of this thing, it has a very fuzzy lumen. Because it has a lot of microvilli to maximize the surface area to reabsorb. So it's, if you are looking at a slice of the kidney and you're going to see a cross section of all these tubules, if you're up in the cortex and you see a tube with a really fuzzy lumen like that, you know you're in a PCT. All right, then I kind of ran out of room here. I'm gonna Okay, so here's my PCT. Reabsorption. The next part is the loop of Henle. And there's the descending limb and the ascending limb. So this was like first described by that guy from the Eagles. No, I'm joking. Don Henley had nothing to do with the loop of Henle here. So loop of Henle that has a descending limb, ascending limb. The descending limb are these thinner, more squamous cells. Ascending limb are more cuboidal. The properties of these loops of Henle are, you are not, I'm going to say it briefly, but you are not responsible on an exam for knowing the details of the properties of this. Basically, the descending limb is going to be permeable to water and is going to be a place where more reabsorption occurs. The ascending limb is pumping salt out, and it's going to, by the time you get to the top of the ascending limb of the loop of Henle, you've pumped so much salt out of the urine that it's actually like only 1/3 as salty as normal body fluids. So this ascending, descending limb of the loop of Henle are doing this kind of magic that actually is creating that osmotic gradient. So this is, remember I said, as you get into the medulla, it gets really salty. The loop of Henle, in particular, I should mention, there are two populations of nephrons. There are cortical nephrons, which are more mainly contained in the cortex. They have, their loops of Henle aren't so impressive. But then there's these what are called juxtamedullary nephrons. They are close, and I'll show you pictures of them. They have their They're closer to the medulla. They have their loops of Henle plunging really deep, penetrating deep into the medulla. And the properties of this loop of Henle are what create that super salty gradient. So again, for this class, it doesn't even matter if you know what it means to be 300 milliosmolar. This is a measure of how salty. It's like 300 milliosmolar when you're up near the top here. It's 4 times. It's like 1200 milliosmolar when you get down deep into the medulla. Up here, you're in the cortex. So as you go down here, it's like, you know, down here is 4 times as salty, basically. And that is going to be functions of this loop of Henle. And we're going to see why that's so important in a few moments. There is then a distal convoluted tubule. The distal convoluted tubule is mostly secretion. So in this distal convoluted tubule here, if there's stuff that's still in the blood, I mean, that you want to get rid of, then you can move it the other way. So distal convoluted tubule, there is something you want to get rid of. You can take it from the blood, from the capillary, and move it into the tubule. So it's going to end up leaving your body eventually. So that's that secretion. Get rid of stuff that you don't want. And then the final tube that we're going to go into is called the collecting duct. And one collecting duct actually can have lots of nephrons feeding into it. And this is going to be, you know, to the renal pelvis here. And again, I'm going to show you prettier pictures in a few moments. I just want to kind of finish the story here this way. So this is going to be called the collecting duct. And this is where the final adjustment of the filtrate happens before it actually becomes urine. Whatever makes it to the very end here is your pee, as you can see. So as this filtrate, though, filtrate started here, reabsorbed most of the water, reabsorbed more of the water, and reabsorbed a lot of the nutrients, secreted stuff. At this point, it depends on the body's needs. If your body needs to hold on to water, let's say you're dehydrated, so you want to make sure that you keep as much water in your body as possible, you want to pull the water back into your body. You don't want it to leave your body as part of the urine. What hormone tells the kidneys that you want to hold on to your water?
ADH.
Exactly. So if there is ADH, what happens is you end up getting these water channels opening up. And if you open up a water channel, another word for the water channel is aquaporin. So if you open up these aquaporins, because it is so darn salty outside, and this filtrate is actually pretty watery. I didn't get into the details, but I was mentioning it's like only a third as salty as a normal fluid. So what happens if you have a really salty solution on one side and it's not very salty at all on the other side? What does osmosis do if water can move? If you take a slug and you sprinkle salt on a slug, what does the salt do to the water? No one ever did that as a kid? I don't feel good about it, but I definitely did it.
It will pull the water.
It pulls the water out. So you know. Yeah, I mean, if you have really salty, the salt kind of pulls the water out. That's kind of the way how I try to intuitively think about osmosis. The really salty areas kind of draw the water. And there's lots of ways to like really think about the physics of osmosis. But to me, that's kind of the intuitive way I remember osmosis is salt kind of pulls the water. So if you have it really salty out here and you open water channels, then the water is going to leave the tubule back into the body. And so you keep the water in your body, keep your water volume up, and you'll have a smaller amount of more concentrated urine leaving your body. So that's the job of ADH. If you don't have ADH, for instance, when alcohol suppresses the release of ADH. So if there is no ADH, in that case, you don't have these water channels. In that case, you have this very dilute urine coming down, and it's really salty out there, but the water can't move, so it doesn't matter. So it just leaves your body as a large volume of very dilute urine. Right? The osmosis can only happen if water can actually move across the membrane of the collecting duct. So that's the job of ADH here. ADH either opens or closes these water channels, these aquaporins, and that allows the body to either reclaim more water, so you have low volume of concentrated urine, or allow a larger volume of dilute urine to leave the body. The one other hormone that works here that we've seen already, what's the other hormone that works in the kidney? Flows from the adrenal gland.
Aldosterone.
Exactly, aldosterone. So let's talk about aldosterone. So aldosterone, which came from the adrenal medulla, that turns on sodium pumps and pulls sodium back into the body. So, and again, this depends on your body's needs. If you're trying to hold on to salt, you're going to have more of this. If you're trying to let go of more salt because you're overly salty, then you won't have as much and let more of the salt leave your body. If your body is trying to really hold on to its water, sometimes you'll release both of these together. If you release aldosterone and ADH at the same time, that maximizes pulling water back into your body, because not only are you pulling water back into the saltiness of the medulla, but you're making it even more salty, right? And you're making by pulling even more salt out of the filtrate and into the medulla, you're driving even more osmosis of water back into the body. So when your body is really trying to hold on to water, it can do that. It can turn on aldosterone and ADH, but they are independent. You can turn them on or off independently to reclaim water or reclaim salt. But again, together they have this synergy. The very last thing we are going to mention is right up here. So if you look, I wasn't just being messy. When I was drawing the DCT, the distal convoluted tubule, I had it going all the way and kind of kissing here at the afferent and efferent tubules. Not tubules, the arterioles. Oop, let's make that red. My glomerulus. So here's my afferent arteriole and efferent arteriole. And oh my God, there is the DCT coming in. And this region right here, both the cells in the arterioles as well as the cells in the tubule are specially modified to be detecting, we'll talk about in a second. So it's gonna be a sensor that helps control kidney function. This is called the juxtaglomerular apparatus. There's special, again, you don't need to know the nitty-gritty details. There are specially modified cells in the arteriole called juxtaglomerular cells. There's specially modified cells in the DCT called macula densa cells. Don't worry about the details as much. What you do want to know is they monitor blood pressure. And filtrate composition. And the basic idea here is to make sure that the kidney function is working optimally. When you go back over to the very first step here, this up in here, this filtrate formation, or just filtration, we could call it, that only works if there's adequate blood pressure. The blood pressure in the capillary is what's forcing the water out. So if the blood pressure is dropping too far, you're going to go into renal failure because you're not making the filtrate. So the kidney is very, very concerned about your blood pressure. The blood pressure is going to determine if the filtrate is being formed at the appropriate rate. And that's also necessary for the filtrate processing to happen properly. If filtrate is happening too fast or too slow, then things don't get recovered enough or they get over recovered. So at this juxtaglomerular apparatus, you're basically checking in on the blood pressure to make sure it's at the optimal pressure to make the filtrate. And you're also looking at the filtrate composition to make sure that it's looking like it's in the target range that tells you that the nephron is functioning properly. And it can do a lot of things to adjust, but let's just say it adjusts the blood pressure and a few other things to make sure that the filtrate formation is optimal. One of its big things that it does can release something called renin if the blood pressure drops. This, again, in physiology, we talk about this a bunch. This is one of the key players in your blood pressure control. When renin is released, it does all sorts of things. It's going to start the whole renin-angiotensin mechanism, and then angiotensin 2 is going to be a vasoconstrictor, brings up your blood pressure, and it actually causes the release of ADH and aldosterone, which are going to increase your blood pressure by increasing your blood volume. So all sorts of stuff. So juxtaglomerular apparatus is monitoring your blood pressure as well as the filtrate and making adjustments to make sure that the kidney function is happening well. And again, one of the most important of those roles it plays is releasing renin as part of the renin-angiotensin mechanism. Again, in physiology, we go into lots of detail. I mean, it's not a bad idea to say renin-angiotensin mechanism. Blah. I'm going to write it out, and I'll tell you why in a second. This is the more formal kind of cascade of events that happens once renin is released. But part of this process of the renin-angiotensin mechanism is this enzyme that's bound onto cell membranes, particularly in the lungs, but in lots of other parts of your body. And that angiotensin converting enzyme is actually what the novel coronavirus uses to bind and inject itself into the cells. So if you didn't have a renin-angiotensin mechanism, the coronavirus would never have a way to get into your cells. So, but if you didn't have a renin-angiotensin mechanism, you would be dead pretty quick because your kidney function would fail and your blood pressure wouldn't be regulated. So it's a good trade-off. But yeah, if you're reading more details about the coronavirus when you're just kind of, following all those papers, you're going to see this ACE enzyme, basically angiotensin converting enzyme, which is bound to the membranes of the cells, is part of this. It's one of the key players in this whole renin-angiotensin mechanism. All right. So what I'm going to do now is just kind of go through some of the pictures in the PowerPoint, which just reiterate what I said, but with prettier pictures. Then show you a little bit of kind of cadavery stuff and microscopy stuff. So let us first, I should mesh voice for you. Are there any questions about all the stuff I've been talking about now? All right, let's share. And let's just start this at the beginning. All right, here's a beautiful little imaging thing of kind of the blood going into the kidneys here. Let's see. All right, so again, this should look familiar here. You've got, whoops, you basically have the renal arteries going in. Let me get my highlighter. Renal arteries going in, renal veins draining out. Oh my God, the celiac trunk, the superior mesenteric, inferior mesenteric, right? You should definitely think about testing yourself. Like when you're looking at these pictures studying the urinary system, it's still worth your while. Like, oh yeah, inferior mesenteric. Oh my God, internal iliac, external iliac. You want to be quizzing yourself because again, the 2.5 weeks is the final, which will include that kind of stuff. Here are the adrenal glands. Again, I was like, most books say like perched like little caps atop the kidneys. And in general, kidneys, anything that has to do with kidneys is usually renal. Like adrenal is above the renal, above the kidney. Renal arteries, renal veins, renal medulla, renal cortex, we're gonna, the renal pyramid. So renal just means having to do with the kidney. as opposed to, what's the one for having to do with the liver?
Hepatic.
Hepatic. Anything that says hepatic is going to have to do with the liver. All right, so this is shown in real life. Here are the kidneys. Here is the aorta. Here's the vena cava. This is showing that they're retroperitoneal. This is a peritoneal cavity. Here is the kidneys sitting against the back wall behind the peritoneum. This is showing it in more detail. This is showing a cut, so a real kidney and a picture of a kidney. So you can see these renal pyramids do actually have this kind of dark look. That's where those collecting ducts are going down. The collecting ducts are all heading down those pyramids and spilling what ultimately is the urine into the calyces, which is then going into the pelvis, which is going to go out the ureter. Same thing here. All of the papilla of the pyramid, again, you know that word now like the back of your hand. It just means nipple-like projection. So the little nipple-like projection at the end of the renal pyramid, that's where all those collecting ducts are spilling out, dumping into the minor callus, with minor calyces come together to become a major calyx. Major calyx all join to become the pelvis. The pelvis then drains into the ureter. So that should look pretty familiar. This word hilus, hilum and hilus are the same word. I think I originally learned it as hilus, so I still always say hilus. But it's just the indented part of an organ that looks like this, where the vessels and stuff come in and out. Like your lymph nodes had a hilus or a hilum. Here are the arteries. So again, the renal artery coming in. Segmental arteries are the first big branches. Then you've got these things here. These are these interlobar arteries kind of going up through the pyramids. The arcuate arteries are the ones that are going along this boundary here. And the cortical radiate arteries are the ones that are coming up. Don't worry about the veins. On an exam, I would ask you about the artery. The veins have the same names, but just to make life easier, just focus on the arteries. Okay, here's a nice picture again. Segmental arteries, that first major branching interlobar going up through the medulla. Arcuate, arcing at that boundary, cortical radiate radiating into the cortex. Got it. This is what I was showing you with that kind of very schematic view of what's going on in the nephron. Afferent arteriole feeding into the glomerulus. Glomerulus having filtration where lots of water and good stuff and bad stuff are getting spilled out as filtrate. Efferent arteriole leaves the glomerulus to become the peritubular capillary, where you can reabsorb the water and good stuff and secrete the stuff you want to leave the body. And this is what is the official term for peeing? You know, you can say peeing, but if you were in a clinical setting, that's not what you would say.
Urinating?
So you don't usually say urinating. if I can draw it with this. Yeah, I can.
Micturition.
Micturition. Micturition is a fancy word for urinating or urination. Often, people often, in a clinical setting, they often talk about voiding the bladder as well. So you're much more likely to talk about micturition or voiding rather than peeing. So this is showing in the glomerulus in more detail these podocytes. Podocytes, those cells that glom around the fenestrated capillaries of the glomerulus to make all these filtration slits. So this is kind of, and this is actually this kind of beautiful electron micrograph kind of showing how these cells have these little finger-like things that interlock to make these little slits that help make this filtration membrane. And again, keep the cells and large proteins back, but allow water and very small things to get out. So this is this process of filtration happening here in this Bowman's capsule. And here's again a picture of it. Under the microscope, you can really see glomeruli really easy. They look like this big glob of capillaries with this simple squamous little envelope around it. So they're super easy to recognize. You're up in the cortex, you'll just see them scattered all over the place. If you're lucky, you can often see a DCT right at the entrance here as well. And you'll know it's a DCT because the lumen is clear. The lumen is not fuzzy. The DCT, remember the PCT, the proximal convoluted tubule that was for reabsorption, has really fuzzy lumen because of all the microvilli, but the DCT is actually pretty clear in the lumen. And again, I'll show you pictures of that explicitly. Just here's just shows you even more detail. The filtration slits, the fenestrations together make this filtration membrane. All right, this is getting more now into the detail. Here's the glomerulus in the Bowman's capsule, PCT, Lupa Henle going down to the medulla, back up, DCT into the collecting duct, which then goes down to the pelvis. Again, that's happening 1 million times. So you've got 1 million of these little nephrons just repeating again and again across your kidneys, working together to do all this processing. One kidney has enough capacity to actually do the balancing you need for your body. So if you only have one kidney, you'll still be okay. That's why you can donate your kidney to somebody who needs it. Although it does mean you have no backup if something goes wrong with the one that's left, or it starts losing its capacity to do its job well. So you're taking a risk. All right, so this is a classic look of a glomerulus. You should definitely recognize, like, I would feel completely fine putting a picture like this on your exam. This is really classic kind of cortex of the kidney. You've got the glomerulus, this big ball-like thing. You've got the squamous cells making this little capsule around it. Like, oh my God, there's a DCT here. You can see because it's got a clear lumen. And these are all PCTs because they've got these fuzzed out lumens. There's like another little DCT there. There's another DCT here. There's another DCT. So when we are done talking and you start just doing like kind of your lab part, you definitely want to look at some slides of kidney or pictures of slides of kidney and be able to definitively say, and not all slides are stained where you can definitively say, I should tell you that. Some of the slides, it's hard to tell. Things get so distorted and there's so many artifacts that you can't really make it out. But in the best slides, you can see it very clearly. This is down in the medulla. Don't worry about the medulla as much. I probably won't show it to you, but what you're seeing here is that the descending limb of the loop of Henle is actually made out of squamous cells to make the tube. So it looks very different, whereas the ascending limb and the collecting ducts are going to be cuboidal. This is more what a real nephron looks like. When I talked about cortical nephrons and juxtamedullary nephrons, cortical nephrons, in fact, they say here, cortical nephron, these are the ones where most of the tubules are up in the cortex. Juxtamedullary nephron, where juxta means near, closer to the medulla, and with these loops of Henle that dip down really deep. And again, this is responsible for creating and maintaining that osmotic balance. So you remember it's 4 times as salty down here. And that is going to be thanks to the actions of these loops of Henle. So in fact, a lot of like the diuretics, loop diuretics are a really common way. Like people have high blood pressure, you want them to pee more. It's gonna, less blood volume, less blood pressure. A loop diuretic works by blocking channels in the loop of Henle. So the loop of Henle doesn't work as well. So it's not as salty down here. So when we open up the aquaporins, not as much water gets pulled into the body here. So that means you're going to pee more. You'll have a higher volume of dilute urine if we don't have it as salty down here. So that's like the mechanism of like those loop diuretics. It actually works at the loop of Henle to keep it from making it as salty down in the medulla here. Don't worry about vasorecta. This is a pretty cool shot. This is like an injection of the little afferent arterioles and glomeruli. Oh, this is showing the juxtaglomerular apparatus. Here you can totally see a DCT kissing right at the base of the glomerulus. Right? You've got this tubule with a clear lumen and it's sitting right here with itself up right up against the glomerulus. So you basically, here's the DCT, here's the glomerulus. And this is actually something you can find. We have slides. If you were in class, we would be able to find these. They're kind of cool. This is showing the same thing, just in bigger, again, glomerulus, classic looking glomerulus, classic looking Bowman's capsule, the squamous cells that make this envelope around it to catch the filtrate. And here is the DCT, this tube with a clear lumen, which is kissing the glomerulus to make the juxtaglomerular apparatus, which monitors blood pressure and other things to make sure your filtration is happening properly. This is just ureter. If you look, it's got muscle and it's got transitional epithelium. We'll look at this in more detail later. In a guy, the urethra is coming out of the bladder, is gonna mix with the seminal vesicles, it's gonna mix with the vas deferens and all. The Cowper's glands feed in here. There's gonna be all sorts of more detail before you actually come out the tip and pee. Whereas in the female, it's much simpler. It's just, here's the bladder, here's the urethra, here's the urethral opening, and you're out. So Again, we'll come back and we'll look at both of these in much more detail over the next 2 classes. Again, this should look familiar just from the first week of class. It's the old classic transitional epithelium with the distinctive, like dome-shaped blobby cells sitting on top. Classic. This is showing that little trigone of the bladder here. Whoa, hold on. Again, for the guy, again, we're going to look at this in more detail when we do the male reproductive system, but you have the prostatic urethra, the membranous urethra, the penile or spongy urethra. There's going to be lots of different sections. So, any comments or questions about all of that stuff? Let's, let me stop that share. So what we're going to do now is look at just, it's a very short thing, but it's worth looking at so you can get a real sense of that adipose capsule of what the kidneys are actually kind of cushioned and anchored in the back of your abdominal cavity there. So I'm going to take us to, oh, where is it? Hold on one sec. There it is. So actually, do you see, what do you see right now?
Just you.
Just me. Okay, I did not do this properly yet. Hold on. What is going on here? Screen. Oh, there it is. Okay, now you see it says cadaver dissections on there?
Yeah.
Okay, so I'm going to click on that. So the first 5 minutes or so are really nice to kind of just show the kidney. And so let's do that. Yeah, I don't know if you've noticed, like if you use their viewer, it's kind of nice if you're trying to save time because you can speed it up.
The last section of the first unit, we're going to dissect the structures along the posterior abdominal wall. So these are all retroperitoneal structures, and let's just get some of the more superficial structures out of the way.
But we actually want to see it bigger, and we actually do want to hear her talk about it. So let's do this.
The last section of the first unit, we're going to dissect the structures along the posterior abdominal wall. So these are all retroperitoneal structures. And let's just get some of the more superficial structures out of the way. I'll flip up the diaphragm and anterior abdominal wall here. And then essentially choose a side to work on and take all the abdominal organs, the gut, and move it off to one side so that you can really get down deep along that posterior body wall. So I've taken all the intestinal tract here. We'll pull the spleen aside. And in advance, we removed a lot of the connective tissue and peeled out the parietal peritoneum here. But what we did.
So, right, normally this would be just under a kind of slippery gossamer film, right? The parietal peritoneum is this thin, slippery layer that normally you would not see this because it's underneath, it's retroperitoneal, but they have taken that kind of slippery saran wrap layer off. And what you're seeing is this thing that looks like a big pillow here.
What we did do is leave the fat that surrounds the kidney in place. And this is a relatively thick layer of fat that helps to hold the kidney in its position along the posterior abdominal wall and also serves as an important cushion for the organ. So if we simply bluntly take that away, you'll see that there's a pretty thick layer of fat around the kidney, even in a relatively thin individual. That will allow us to expose the kidney itself and dissect it. Now, as you remove that fat, be careful not to also remove the adrenal gland or suprarenal gland that sits just at the superior pole of the kidney. It does look a lot like the fat tissue. It's a little bit more orangey in color, but do be cautious not to remove that as you pull away that posterior fat. Now, the first thing that we're going to do is examine the kidney and section it to take a look at some of the internal growth structure. Using the large knife, you can make a frontal cut through the kidney, essentially cutting it in this plane, and then you'll be able to open it like a book and look at some of the internal structure. Now here at the hilar region of the kidney, we can see the renal artery penetrating the organ. We can see the ureter exiting the organ. At the moment, we can't really visualize the renal vein that would be draining blood away, but let's take a little look at the tissue of the kidney itself. It's separated into an outer cortex that looks rather granular, and then an inner medulla, which is organized into renal pyramids. And I think you can make out the shapes of these renal pyramids even grossly. Now, after the urine is made within the cortex and modified within the medulla, it's going to drain into the collecting system, ultimately to enter the ureter and be stored in the urinary bladder. So here we've got part of this collection system where we've got a structure called the minor calyx receiving urine from each one of the renal pyramids. Here's a nice example of a minor calyx. Here's the tip of another renal pyramid sending its urine into a minor calyx. The minor calyces will combine with each other to form a major calyx. Ultimately, all those major calyces will drain into this upper dilated portion of the ureter known as the renal pelvis. From there, the urine will pass down through the ureter and be stored in the urinary bladder. Now we're going to look at the vascular supply of the adrenal gland, and it is an endocrine organ, meaning that it delivers secretory products through the bloodstream. So for such a small gland, it's got a pretty excessive, or extensive, I should say, blood supply, with the vessels supplying the adrenal gland coming from above, from the medial side, and from below. Okay? And these are, more specifically, branching from the inferior phrenic artery, which supplies the inferior surface of the diaphragm.
Yeah, so this, we don't care about that so much. Let me close this.
The branches coming down to the adrenal gland from the inferior phrenic are the superior.
So that just kind of gives you a nice view of what the kidneys actually look like and how they are kind of really embedded in a huge amount of fat. When you first, again, go behind the peritoneum and think, oh, now I'll see the kidneys. It's like you just see like a pillow of fat, and it's kind of, you have to kind of get into that pillow of fat to find the kidney. The other thing I have, let me find, give me one more moment. Oh, I know where I want to go. Okay, let's go. Give me a second. Modules. Okay, let me share again. Share. Where'd it go? There it is. So here I'm in just this histology guide again. And if we go into the urinary system, you know, and you just like look in the kidney. When you first look at a slide like this, you can start zooming in and just see all of these little glomeruli everywhere. Do you see them scattered? You know, so that, I know this is the cortex because it's just full of little glomeruli everywhere. You can just see them, the little balls. You know, as I go down away from there, yeah, then I'm getting into the other parts. Let's stick around, let's stick in here right now. As I zoom in more and more and more and more, again, a glomerulus, the squamous capsule that goes around. This one isn't stained as pretty, but you can still make out, like this thing that I'm doing the little hand with would be like a DCT because it's relatively clean. Whereas some of these other things that are really just kind of messy and fuzzy inside would be PCTs. Although this isn't as clear. Oh, over here, you can see this is obviously cleaner than some of these other things that are super fuzzy. Like this is not clean. This is pretty clean. Over here, this is pretty clean. This is not clean. So, you know, as you're looking at this stuff, you know, on an exam, I would do my best to find one of these that is going to, yeah, have a real obvious, like this is DCT, this is really PCT. You know, you can tell. This puppy here that I'm doing the circling with the hand is got a clean lumen. This thing here, the lumen is really not clean. It's all full of long microvilli optimized for reabsorption. So that's PCT. So, you know, and then obviously whenever you see something like this, you know, you're in a blood vessel because it's full of little blood cells. So this is parts of a little vein right here. Actually here you can also see other vessels full of little, yeah, it's always cool to find all the little, realize, oh yeah, there's blood cells everywhere because everything has to be penetrated and permeated with blood vessels and capillaries. If I look around, usually if you look around a little bit, you can find a juxtaglomerular apparatus, although I can't promise you. You know, that picture that I showed you in, you know, from the Marib text was, you know, was about as pretty as it gets. But just again, to say, if you see a tubule that looks pretty clean versus a tubule that is so not clean in terms of its lumen, you should be able to kind of start to make that distinction. Let's look at another one. Okay, this is basically the same thing. Again, clean lumen, fuzzy lumen. What about this one? Oh, I can't say I like this stain that much. What else? You know, and in terms of, you're not going to have to recognize, oh, I'm in the bladder or the ureter, but you should definitely, just as your, you know, basic knowledge, your basic toolkit of tissues that you are still responsible for. If you go and look under your virtual microscope during your virtual lab practical and you see tissue that looks like this, you know, you would say, I'm in the urinary system, urinary tract someplace. Like nothing else has those big dome-like globby cells like stacked on top in a stratified kind of thing like this. You know, don't worry about, am I specifically in the bladder or in the urethra or something. They both are basically stratified. They're both transitional with smooth muscle underneath. You know, as we go underneath, it's smooth muscle. So this, I mean, this should all look like smooth muscle. Like If you were here, you would have no idea if you're in the wall of the intestine or in the wall of the bladder, right? Smooth muscle lines the walls of most of your hollow organs. We're going to find it again in next week when we look at the uterus. The uterus is just full of smooth muscle in the wall. So, your basic kind of tissues, hopefully it's kind of fun. By this time of the semester, you can look at this and it's not scary anymore. It's like, oh yeah, there's smooth muscle. That's cool. Just to kind of show you why it's called transitional, right? If you are relaxed, then you get all those big globby looking cells, those dome-like cells on top. They stack up all weird. But if it's stretched out, let's say it's like, oh my God, I gotta pee, find me a bathroom, please, fast. And again, your bladder can hold up to about 1 liter before it's gonna start leaking out. Here we go. You can see it almost looks like stratified squamous. It's got this much flatter looking top. So I am not gonna do that to you. I am not gonna put stretched out transitional epithelium on the test trying to, catch you or something. So, but just kind of making it clear why. What is this? How about, what's this thing right here? You should recognize this thing that I'm circling around right now.
Is that an artery?
That's totally an artery because it's got like thick, smooth muscle in its walls. Whereas if we look next to it, come on, reload, Mr. Page. Oh, come on, reload. Oh, now I don't know why my internet is slowing down. Okay, yeah, and then if you look kind of right next to it, this is totally a vein because it's got these super thin walls that are almost like a capillary. It's basically like simple squamous epithelium with some connective tissue practically. Whereas an artery has very clear, thick tunica media with smooth muscle. And here's classic just smooth muscle looking like total smooth muscle. So that's the other thing you can do when you're looking around for other things. Make sure that you're still remembering all these. You can review as you go along. So any comments or questions or anything about all of this? Let me stop the sharing.
I have a question for the intestine and like all the layers. For the muscularis layer, does it have to be specific which one it is?
No, for the urinary system, you should just know that there is smooth muscle under the mucosa.
Okay, thank
you. Any other questions or comments or anything?